Yes.
Now okay.
Okay.
Welcome everyone out there who's watching from remotely. We had to take care of a few last minute tech issues, and now I think we have everything squared away to begin. So thank you for your patience. My name is Gene Van, and I'm teaching AP Environmental Science here at the Royce School. And this past week, we've been working with the Headwater Sciences Institute in helping students to develop an experimental question, to gather data on it, to analyze that data, and now to present on their findings. And so I'm looking forward to seeing what the students have produced from their work. It's been an interesting week, and we have an order. By the way, folks, I put this up so that it could more easily, the camera could see your face. Bryn, did you want to say a few words before we let the students take over?
Yeah, I'll just say hello. My name is Bryn. I'm with Headwater Science Institute. I'm the program manager, and I've been working with this class at Royce over the last few weeks to develop these projects. I'm also super excited to hear their presentations. They put a lot of hard work into collecting their data and doing their statistical analysis, and I'm sure they'll be just great. So if the first group wants to come up and start presenting, we'll get started.
All right, so here we go. Give everyone a hand.
So wait.
And it looks like we need to project this so y'all can see it in the classroom. Pardon me. I thought this was already projecting. Okay, there we go. All right, thanks. Sorry for the interruption.
So wait, do we just stand in front of the camera?
Yeah.
Okay. Okay, so guys, come here. Okay, so you can start us off. Okay, yeah.
Hi, I'm Annika.
You gotta go in the camera.
Why? We're all just
Because it's a record.
Just go in.
Okay.
Oh.
Okay, ready?
Go ahead, Isabel. Introduce yourself.
I'm Isabel. I'm Kayla.
I'm Annika.
And I'm Ian.
And we tested the effect of human restoration on dissolved oxygen levels at Salsa Creek.
Okay, now you can go. Next slide, please.
Changing
it? Do we change it or does she change it?
Good question.
There we go.
Okay.
Go ahead.
First, we want to start by defining dissolved oxygen, which is the amount of oxygen available in water to living aquatic organisms. So why should we care about dissolved oxygen in water? Aquatic species depend on oxygen-rich water, and without sufficient oxygen, they will not be able to survive. A small change in dissolved oxygen concentration can affect the composition of aquatic communities and really affect habitats. So we tested at Salsa Creek, and what we did is travel to Salsa Creek in Oakland, where we tested the impact of human restoration and activity on dissolved oxygen levels in restored, unrestored, and undeveloped areas.
Okay, so for some background information on restoration in general and then the Salsa Creek Restoration Project. So site restoration is the repair and treatment of land and bringing it back to its prior condition, which most commonly makes up for human damage. And this is a positive form of human impact on natural areas. So the picture below is the Salsa Creek Restoration Project, which happened in 2015. And so the goal of this project was to create a sustainable creek and park environment for park enjoyers, fish, and wildlife. And the project included thousands of newly planted plants and trees, over 250 feet of removed culvert and concrete to open up the underground buried creek, improved flood capacity, removing obstructions in the creek, daylighting and increasing the area available for water to pass through the channel, and then finally increasing water storage capacity, which reduces peak flows and increases flow duration.
Okay, so the question that we were trying to ask ourselves in this is, what is the effect of human restoration efforts on DO levels in Salsa Creek? We have an independent and a dependent variable. Our independent variable was the human restoration areas, and our dependent variable was the dissolved oxygen levels. So our hypothesis was that the areas of Salsa Creek in which the human restoration efforts, that includes increased planting, restored natural habitats, increased flood capacity, stability, and increased shade, which were all the goals of the restoration project in Salsa Creek, will have a higher dissolved oxygen level in the water due to a more eco-friendly environment. Our null hypothesis is that the areas of Salsa Creek in which human restoration efforts were put in place will show no difference in dissolved oxygen levels in the water.
Okay, so when we got to Salsa Creek, we used our thermometer to measure the temperature of water in a specific area. We then identified the turbidity of the area of water being tested. We then inserted our DO monitor into the same area we tested the temperature in to test the dissolved oxygen levels. We cleaned the monitor with distilled water and repeated this process 2 more times in the same area to collect multiple points of data. We repeated steps 1 through 3 for each of the 9 areas we tested. So the materials we used were a dissolved oxygen meter, a thermometer, distilled waters, the SparkView DO app, and a camera. Okay, so for our restored area, we went to 3 different sections, and within each section we tested 3 areas. So the first one was on one side of the culvert. These are exact pinpointed locations. This is our second section, which is a little bit farther away from the culvert. Here's Ian taking our measurements. And then our third one was even farther away, and the 3 pinpointed locations.
We put all of the data that we recorded into a chart where you can see for each of the 3 sections and each of the 3 areas per section, we found 3 different data points based on trials for temperature and dissolved oxygen. And then we found the averages of each, as well as recorded the turbidity for each site. And we were able to put that into a graph.
Okay, so we then measured unrestored areas, and this means upstream of the culvert. And these are areas that haven't been restored and touched by human development and by the Salsa Creek project. As you can see, our first section of the unrestored site was right outside the tunnel, which is the culvert. And you can see the pinpointed locations. We decided to move up the stream through the areas. Go to the next slide. Yeah, so this was our second section. This was farther upstream, still in Salsa Creek. Took data, and these are the exact pinpointed locations. We can go to the next slide. So we then decided to travel outside of Diamond Park up near Joaquin Miller. And that's where we did our undeveloped site, which is kind of part of our unrestored section. We took 3 data points in these areas. We traveled to 3 different sections of the undeveloped site and measured data.
We then used the same process as we did for the restored site and put all of our data into a chart based on the 3 sections, the third one being the undeveloped site, and found the averages for our temperature and dissolved oxygen trials.
And so here is what our evidence looks like. As you can see, the first graph on the top left measures the dissolved oxygen levels and the 3 sites and 3 sections that we looked at. So as you can see, the restored areas had significantly higher dissolved oxygen levels on average. The unrestored had lower, ranging from 7 to 7.5, and the undeveloped, we did have lower data sets, so that's why there's only 3. But there was a greater range and therefore greater error bars. In the restored and unrestored areas, the error bars do not overlap, which means that there is a significant difference between the 2 data sets, which is a good thing. And the unrestored and undeveloped areas do have overlap, which makes sense because both of them are not restored, and so you would think to see more of a correlation between the 2. On the bottom right, we can see the effect of temperature on dissolved oxygen levels. This is contrary to our hypothesis. We thought that with lower temperature, there would be higher dissolved oxygen levels, but from our data, we found that with higher temperature, there was higher dissolved oxygen levels. From the captions, we can see that we ran a statistical Anova, and that there might be a relationship between the temperatures and dissolved oxygen. Our calculated p-value was 0.14. And that indicates that our null hypothesis was slightly ridiculous.
Okay, so what were the main patterns of our data? Higher temperature correlated with higher dissolved oxygen levels in the respective areas, and lower temperature correlated with lower dissolved oxygen levels. Measurements from the restored area had higher dissolved oxygen levels, and the error bars in the dissolved oxygen graph show no overlap between the restored and unrestored areas. This makes sense because the two areas have different ecosystems. However, the error bars show overlap between the unrestored and the undeveloped areas. This result makes sense because both areas had no human restoration efforts in place. So how did our results compare to our hypothesis? There was a significant increase in DO levels in the restored areas in comparison to the unrestored and undeveloped areas. However, in contrast to our hypothesis, we found that the areas with higher temperature and less shade had more DO levels rather than less. So what do we think? What do you think you saw the results you did? According to the report done by the City of Oakland on the restoration efforts on Salsa Creek, they plan to daylight New Creek and regrade existing creek banks to a more gentle slope. Plant thousands of new and native plants and trees, reconfigure the creek to improve flood capacity, create stability, and restore natural habitat. The daylighting of the creek most likely contributed to an increase in the area available for water to pass through the channel, increasing storage capacity and flow duration. The increased amount of planting in the restored areas most likely contributed to an increase in photosynthesis, which would produce more oxygen in the water.
Okay, we can go to the next slide. Okay, so the question is, how do our results compare to current science and the biological context of the field site? We actually decided to do some outside research on other restoration efforts in other parts of the country. We decided to look at Tampa Bay, Florida, and some efforts that were done in Alligator Creek. And through their study, the EPA, which is the Environmental Protection Agency, showed a 1.3 milligram per liter increase in dissolved oxygen levels through their restoration efforts. Comparing that to our results, Salsa Creek showed a 0.5 milligram per liter increase in dissolved oxygen levels. Both show an increase. However, obviously Salsa Creek did not show as significant of an increase. Yeah.
So how could our research affect our understanding of the ecology of the area that we researched? Now that we understand that the restoration efforts have positive effect on DO levels in the restored areas of the creek, this means that now we can make more effort to restore other stretches of Salsa Creek that might need more work done. What could our results tell us about ecosystems like this one more broadly, especially when thinking about global environmental health? We tend to think of human influence and intervention as always bad for the environment, but our research actually shows that in certain cases where humans attempt to restore and reestablish struggling rivers and creeks, the results can even trump areas that have been untouched. With lots of hope lost in the reversal of global warming, our project shows that positive environmental change is possible and action should be taken. Other urban creeks looking to improve creek conditions should consider these projects, since restoration seems to result in higher DO levels.
Okay, so looking at future research based on our findings, we would look at the impact of turbidity within any type of water, not only creeks and streams, on dissolved oxygen levels. So turbidity is the condition resulting from suspended solids in the water, including silts, clays, waste, sewage, plankton, and these particles absorb heat in the sun, which raises water temperature and then results in lower dissolved oxygen levels, which we also concluded. So then from what we learned, we would most likely want to increase the sample size just to maximize the precision of data and our results for future experiments. And this is our bibliography. Thank you.
Thank you.
I want to leave an opportunity for questions.
Sure, you guys got a meeting.
And
Stop.
folks who are watching remotely, I believe there's an opportunity. Is there one more slide you all need to show?
Thank you. Yeah.
Okay, there we go. So let me get out of full screen. So if there's anyone at home that wanted to ask a question or watching remotely, you are welcome to type one in. Anyone in the audience, a question for our DO presenters while we're waiting? Any questions at all? All right, well, I'll ask a question. I was curious to hear, what was the main, I know you covered a bunch of factors that the restoration, how it may have impacted. Is there one particular feature that most likely contributed to the higher dissolved oxygen levels in the restored area versus the unrestored area?
I think that the planting was probably the biggest effect. I mean, if we think about like the role that plants play in like getting oxygen into the water, like algae, and we talked about like other forms of planting. I mean, they did significant planting. They said like thousands of new plants, and I think that the other aspects of the restoration didn't have direct effects on oxygen, but I think that just increasing the number of plants, therefore increasing the number of oxygen, probably had the most significant impact.
Okay. All right. Wonderful. Thank you, guys. All right. We'll let the next.
We have some questions coming in from online. So,
All right. Let's hear it.
this question here: would a change in weather affect the findings
or results?
All
right.
Do we get this one? Does anyone want to answer this? No one? Okay. Yeah, I mean, obviously in cloudy weather that affects the temperature. As we saw that like temperature did have an effect on the DO levels. And so with colder temperatures or hotter temperatures, there's obviously going to be like varying levels of dissolved oxygen. I would say that rain, I'm not sure. I mean, I think that would that increases the amount of water in these streams and the creeks. So obviously, I think that correlates to higher DO levels. But that's not something that we tested. It was pretty shaded, so I don't think that rain would have a significant impact on the DO.
And it's even fair to say this would be a great question to do further research on.
Yes.
Even if if you're not certain. So, yeah. All right. Hey, thank you, group, for going first. Thanks for testing out this platform. Thank you all at home for watching. You're watching some learning in progress too. So our next group is is he? Well, let's see here. Jen, is it possible to queue up the next presentation? We had someone who had to use the restroom for a second. What's that? All right. So the Bug Boys group is next. Yes, you were going to go third, and then we were told the next group was missing someone, but now, all right. So can you all get started, and then we'll assume your group member will be back here. Actually, all right. That did catch that we just had a few people. Do y'all mind? Okay, thank you. All right, so we'll let's give a hand to our Bug Boys group. All right, and remember, folks, hey folks, remember we're live. Okay, voices carry. Okay, and I am paying attention to the audience as well. Okay, so let's have the Bug Boys get started. Excuse me, I'm sorry. You all call it yourself.
We can go with the insect individuals. That works too. All right. See, it's gonna tell us where to go.
Go ahead. You take it away.
Hello, we are the Bug Boys group. Weird name, but we were researching the effects of human development on insect number and variation.
Hi, I'm Audrey.
I'm Duncan.
Charlie.
And yeah.
So, introduction. Insects play crucial roles in the long-term health of environments, from pollinators to detritivores. Thousands of species depend on healthy insect populations. As humans continue to shift climate conditions and alter ecosystems, insect populations are also shifting, with species becoming at risk or disappearing entirely. Many sources agree that we must act now, or we could be at risk of a massive upheaval in our ecosystems. As insects are an often underappreciated aspect of an ecosystem, I know a lot of people have some fears about bugs, we decided to focus on them and their impact on Salsa Creek. More specifically, whether an environment less affected by pollution/human habitation would lead to a higher amount and variety of insect species.
So our main question was, what is the effect of human development on insect quantity and variety? Our independent variable was the proximity to human frequented areas, and our dependent variable was the amount and variety of insects categorized by flies, termites, beetles, worms, and spiders. Our hypothesis was that the less affected an area would be by human development, the more insects we would find. And our null hypothesis would be that the less affected an area would be by human development, the less insects we would find.
A slight addition to that, I think we made some changes that may not be quite reflected on these slides. Our hypothesis was additionally that the less affected an area would be, the more insect variety we would also find.
So to do that research, we first entered the creek through a path made by humans, traveling further down the creek through both secluded and non-secluded areas. We used insect nets, tape measures, and recording materials for our data. And with those nets, we swiped our nets 10 times to have 3 replicates per area and measured how close a human developed area was. And that could be like walking paths, sewers, parks, and houses. And we did this for 11 areas. And we also considered whether the human developed area was helping environment or not, for example, creek restoration. And we took notes on the number of insects and the types of insects we found.
Right, so here is our evidence compiled into a graph. This is for total insects versus like meters away from human habitation. Our p-value ended up being 0.09, which indicates that there is a connection between the meters away from human habitation and total insects. And our R-squared value was about 0.209, also indicating that there is some connection. However, it's not a perfect match for that being a super strong correlation. But yeah, there's definitely some evidence for it. Oh, okay. This one is for our other hypothesis, which was that species variation would go up as we went away from human habitated areas. And this one was pretty much entirely off. We had no correlation between the two. We found a large number of species close to human habitation and a large number of species away from human habitation. So our p-value was 0.51, which indicates no correlation. And our R-squared value was about 0.025, I think. So also very little correlation.
Okay, so for our discussion questions, so we first asked what were the main patterns in the data, and as we expected, the further away we got from the human development/pollution, the greater numbers of insects we would see, and that was supported by our data. And then when we went closer to human developed areas, there were fewer. So, however, there's no real trend about diversity. And then how did your results compare to your hypothesis? They supported it for the bug population, except for the variation. And so this included when we went to trails and cleared paths, and then we were far away from houses and parks and trash. But yeah, why do you think you saw the number of results you did? The number and diversity of insects are declining about the globe, around the globe due to habitat loss, pollution, and climate change originating from human impact. So that's why we greatly focused on the human impact of Salsa Creek. And so thus the areas of Salsa Creek with more human impact would be expected to have lower diversity of insects. Things like human trash and physical disturbances would just discourage insect settlement in the area as well. So that's why we also added population. So how do your results compare to current science and biological context of the field site? Our results supported the findings of the Society for Conservation Biology, as they note that habitat loss and degradation Pesticides and climate change are the most influential factors of insect declines. Even though Diamond Park is a great place for humans, it's not necessarily like we're not necessarily harmful to certain areas of like natural ecosystems there. And then for part 2, how could your research affect our understanding of the ecology of the area you researched? So while Salsa Creek has areas that have been negatively impacted by humans, our group was able to locate multiple areas with little impact that showed signs of population diversity. We believe that this is due to the care the community of Oakland has put into maintaining and restoring the creek's ecosystems, as well as the negative impact human population has had on insect habitation. What could your results tell us about ecosystems like this? One more broadly, especially when thinking about global environmental health. So with an estimated 5.5 million species of insects, which is crazy, the most diverse animals of like on the entire planet, more than 1 million have been named by scientists, and many more have yet to be discovered. In fact, insects account for 80% of animal life on Earth. However, without widespread action, many of these important creatures in places like Salsa Creek could face extinction within the next few decades.
So looking towards the future, we saw some factors that we didn't necessarily get to account for that we really, really wish we did. We were going to maybe talk about plant species and how those affect insects. For example, in this picture of berries here, we saw a lot of this particular type of black fly gathered around the berries. We thought maybe it had to do something with the flies sucking the berries' juice, but we really didn't get a chance to go for that. We wanted to talk about invasive species and what would cause a prevalence of one species over another species. What are some ways we could get a maybe more accurate number of insects in the location? As much fun as our swiping bug nets was, maybe not the most accurate way to gather information, and we'd like to do a more thorough research, have more sample sizes, that sort of thing. We want to talk about how water quality affects insect populations, water pH, amount of dissolved oxygen, all those sort of things, and maybe how they affect aquatic bugs. How does air quality affect insect populations? Is air quality tied to being closer to a road or to a populated area? And how would that affect our insect populations? And what are some ways that we as humans can help insect populations recover? Would that be planting more types of plants? Would that be creating better habitats? Like, how could we do that? So in the future, we're going to attempt to get a bigger sample size, as our 11 sample size is maybe a little too small, and think of ways to better identify insect species. Thank you so much.
Thank you.
Thank you. Appreciate it. And thank you all for being willing to go second, even though that wasn't your original order. So any questions from the audience? I'll ask a question. And let's, here we go.
Ooh.
Let's take one.
If I could change one thing, I think I would want a bigger sample size. 11 was just too small. I think we needed a bigger sample size, but at the time we didn't really consider that.
And I'm interested in knowing that app that you used to identify the bugs. Did anything interesting come up?
Yeah, I mean, we got a ladybug once, but unfortunately the bugs are small and they fly away very quickly, and we didn't get a whole lot of information just because they were so quick. Also, I really didn't want to like touch the spider enough to like hold it up to the camera. But yeah, maybe in the future, if we brought containers or some way to temporarily house them so we could get the species, that would be better.
Okay, great. Well, thank you all very much. Appreciate it.
Thank you.
All right, so the nitrate group is next. That's great, wonderful. All right.
Hi, this is our Headwaters research project. My name is Lucas.
I'm Vincent.
I'm Sage. I'm Jeffrey.
All right.
No, no, no. Wait, Sage. I deleted your fucking.
What?
Okay.
There we go.
Sorry about that. Tech difficulties.
All right, so during our project, we studied the effect of human development on nitrate levels in Salsa Creek. So we decided on our testing locations by taking into account the surrounding environment. So at the upper sections of the creek, there was little to no human impact, whereas at the lower sections, there were houses on both sides. We knew that factors of high nitrate levels could be runoff or leakage from fertilized soil, wastewater, landfills, animal feedlots, septic systems, or urban drainage. Therefore, our hypothesis was that areas with higher human development would have higher levels of nitrate in the water. So at the creek, we tested different samples of water in a restored site, a site next to a lawn, a site next to a playground, and sites further up the creek with little to no human impact.
All right. So our first site was the unrestored site in Diamond Park. So we went down next to the culvert and tunnel and then measured the water. And our next one. Oh. The next one was the restored site in Diamond Park. So down below the tunnel and culvert. And then the third site was at Joaquin Miller Park. So this was the completely undisturbed site because Joaquin Miller's like a protected park. So there was no like runoff from houses, or there shouldn't have been any. Yeah, next one.
All right, so our research question was, what is the effect of human development on nitrate levels in the water? Our independent variable would be human development. Our dependent variable would be the nitrate levels. And as we said before, our hypothesis was that because of human development, the nitrate levels would be higher in higher developed areas. All right, for our materials, we had a nitrate test kit, which had a vial, nitrate, 2 different nitrate tablets, an octo slide comparison device. Next, we had writing utensils, a journal to like write down our data and everything. And we had timers to like time the test kit. Yeah.
All right, so our method was to use the nitrate testing kit, which consisted of the materials listed to collect our evidence. So first we used a vial to collect 5 milliliters of water from the creek. Then we put in our first tablet and shook the vial until the tablet dissolved. After that, we put in our second tablet and again shook the vial until that one dissolved. Then we set a timer for 5 minutes, and after the 5 minutes were up, we compared the color of our solution to the colors of different nitrate levels using the octoslide comparison device.
All right, so what were the main patterns in our data? The main pattern showed that the nitrate concentrations changed according to the location of the water. So this is a pool or a riffle, and they also changed even more drastically the closer it was to human development, so upstream versus downstream. However, in our graph, the error bars overlap, meaning that we cannot say for sure that there is a distinguishable change. And how did our results compare to our hypothesis? They were actually on par with our hypothesis. And why do you think we saw the results? We saw the results because the closer to human impact, the more nitrate runoff there will be. Contrastingly, the further away we are from human impact, the more diluted the nitrate will be as it settles and thins out along the water. Our results match current science, which notes that nutrients such as nitrate dilute downstream. A study measuring downstream changes in nutrients in an Ozark stream shows that nitrate is less downstream than upstream in a variety of locations and times, which matches with our findings. How could our research affect the understanding of the ecology of the area? Our research could affect the understanding of the ecology by showing us the impact of human development of the water, on the water. And our results tell us that ecosystems like this one are impacted by human development. So we must be cognizant of the impact we are having on wildlife in order to ensure its well-being.
Next slide. All right, so here's our evidence, our results. So the graph shows the relation of nitrate levels in human developed areas versus non-developed areas, as we said. The graph from the study shows that because the error bars overlap at certain why is it? This is like the internet was Oh, okay.
Hopefully, it looks like things are paused on the camera there, so that hopefully,
it's on that.
if they're seeing it remotely, right now we're stuck on the discussion screen, but keep
Okay.
talking.
All right. So basically, basically on our graph, when it shows up, if it does, you'll see like error bars, and these basically are to tell us how accurate our data was and how much it could vary. Like the values could vary. And what it showed was that from the sites like next to the park and the Riffle Site 1, where there was like large human development, there were higher levels than the last site, like the Joaquin Miller site. And yeah.
It may just be the Apple TV, so I would bet folks who are watching remotely are able to see it. So
All right.
let's assume that.
All right. And then for future research, so based on our findings, we thought it might be beneficial to look into the different drainages that feed Salsa Creek, as that would inform us on where the majority of nitrate originates. And then since we've learned that there's a correlation between human development and nitrate levels, it would be interesting to like further research the environmental impacts of nitrate contamination in the creek. And then potential questions include, what is the impact of nitrate levels on What is the impact of nitrate levels on animal biodiversity in creeks? What is the impact of nitrate levels on pH levels in creeks? And then how do increased nitrate levels impact plant life in creeks?
All right. Any questions?
Any questions from the audience? Or from home?
Is there a question that pops up?
No, there's no question.
They said yes. Oh,
Let's see.
I don't
if
think
there's
there are any
one.
questions. All right, take it back.
We could see it the whole time.
Have a question?
So
Yes. Okay.
Going back to the question we got in our previous presentation, if you could change one thing about your experiment, what would you?
Well, like you guys said, like we had limited time to test and we didn't get that many samples. Like we only got to test like 3 at each site. Also, like we didn't have enough tablets to test as much as we wanted. So like definitely more time and more sample, a bigger sample size. Yeah.
All right. Let's give them a hand. Thank you guys very much. So we have just a quick time check, folks. We have 20 more minutes, 2 more groups to present. So we just want to be mindful of the time. And what I'll do is we'll try to aim for 9 minutes in your presentation. And then I'll just give you like a 1-minute warning before then. Does that sound all right? All right. So let's have our tree folks come up. Oh, we just missed a question. So do you all see this question on the screen? Let me see if I can get it to appear for you. Let's try this again. All right. Is it appearing now? Let's see. Okay, it looks like Apple TV is not working. Let's see if we can get this to turn back on again. Excuse me, folks back home, we're having an issue with our Apple TV.
Hi, Jean. For time's sake, let's just go ahead and have the next screen come on, okay?
Try this one more time. Okay, so have to do this the old-fashioned way. For the benefit of our folks in the classroom. Thanks for your patience. Can't seem to find my dongle now. All right.
Jean, are we able to move on with the next
I have a
group?
feeling there may be some, we can talk later about why those might have shown. Okay.
Can you still see it
Change
on your
this
computer screen?
here. let's change this to inputs. Okay, thanks for your patience, audience, and the classroom now can see the presentation. So we'll go ahead and let folks begin. So thank you for your patience. All right.
Okay. I'm Nicole.
I'm Rachel.
I'm Eleanor. We did Which Tree Will It Be? The Effect of Tree Type on Soil's pH. So the pH of soils surrounding different types of trees differs based on the specific species. They grow where they do because the climate and the soil are suitable. So there are some other potential effects on pH, such as rainfall. Rain is naturally acidic, so if there was a lot of rainfall, the soil would have a lower pH. There's also runoff. Trees exposed to potential runoff will be affected by the pH of the runoff. And trees with the higher nutrient uptake will have more hydrogen ions, as a result, lending towards a lower pH. And trees with lower nutrient uptake will therefore have fewer hydrogen ions and will likely have a higher pH. So we should care because the soil acidity could affect the health of the detritivores that live in it. So ensuring the soil is at the proper pH to ensure a stable environment for them is very important.
So our research question was, how does the pH of soil vary based on the area or type of tree it is or is at? So we actually ran a few tests that you'll hear about a little bit later. So we had two independent variables. One was the trees that were in the different restored areas. There were 3 levels that we did: restored, unrestored, and lightly restored. And then the category of the tree was the other independent variable, and then we were measuring the pH level of the soil. So our hypothesis was the pH of the soil will be consistent throughout the different species of trees and not throughout the specific areas. So our null hypothesis was that the pH would be consistent throughout the area and not throughout the species of tree.
Okay, so the materials that we used were a little thing to measure the pH and like the little pill capsules that come along with it. We used 2 different transects, the Seek app to help identify types of trees, distilled water to make sure that the pH wasn't impacted by the water, a spoon, a dropper, and gloves. So basically what we did was we designated 5 10-meter long transects in 3 different locations. The first location was moderately impacted by humans, the second was a restored area, and the third had little to no human disturbances. At the beginning and end of every 10-meter transect, we measured the pH of the trees that lay within 4 meters on either side. When testing 2 different transects in a single location, we set them 50 meters apart to ensure that we didn't have like biased data.
Okay, so this was the results from our first test. So this is just the 3 areas that we split up the trees into. We did transects at each of them. The control, we didn't get to do as many samples, which is why there's only 2 data points. So that could be a reason why our data was not super accurate. So at first glance, it kind of seems like the restored had a higher pH than the other trees in the other areas. But we actually ran a test and you'll see that that's actually incorrect. But as far as the locations, they're pinned on the map. We did 2 at Salsa Creek / Diamond Park, and then the other one was by Joaquin Miller. That was our control. So they could be differed by that as well. Okay, so these are the statistical Anova tests that the results that we ran. So we ended up getting 3 different p-values based on each of the sections that we tested the trees in. So our sample size was 19, again, not that much, which could be a reason behind this. So we got p-values that were above 0.5, which means that we failed to reject the null hypothesis. So our data didn't really prove anything, even though at first glance it might have seemed like it could, just based off of the graph, and you could kind of see that the pH was a little bit higher in the trees that were in the restored area. the p-values proved that this was inaccurate. So we were not able to prove our hypothesis correct. So then we also did a second graph. And this one was where we looked at our other independent variables. So instead of looking at like the different areas, we looked at the different types of trees. And as you can see by looking at it, there was no significant difference between coniferous and deciduous trees. If we had more data, we would be able to come up with a better conclusion. The p-value was 0.38, indicating that there's a 38% chance that we could reject our null hypothesis. And yeah, you can also tell by the overlapping error bars.
So there were some patterns throughout our data. All of the soil ranged from 6, which is acidic, to 7, which is neutral pH, with the average pH being 6.5. The deciduous trees and trees in the restored area appeared to have higher pHs. And this compared with our hypothesis because our data did not support our hypothesis because our p-values were way above 0.5, meaning we failed to reject our null hypothesis. We think that we saw the results we did because there's no significant difference between the type of tree area and the soil pH, likely because it has more to do with the proximity to the water and just the more general location. Soil that is near water will be influenced by the water's pH more so than a uniform pH based off the type of tree it is.
So then looking at how our results compare to current science in the biological context of the field site, the city of Oakland has efforts in place to decrease erosion and improve water quality in Salsa Creek, meaning there could have been a correlation between the lower pH of the water and the results we got. How could your research affect our understanding of the ecology of the area you research? Our research displayed that the pH of the soil was not heavily affected by influences such as location or tree type. Sure. Wait. Okay. This one? Yeah. So for like the more broad factor, we were thinking that the results tell us that the pH of soil is a constant in our environment that's less dependent on external factors. And the soil pH may be determined by the nutrient needs of the tree. So each tree kind of has specific things it needs to grow, and so it takes up soil based on that. So some trees might require more nutrients, and therefore there'd be more hydrogen ions, leading to a lower pH in the soil. So in terms of future research, it would probably be good to ask. It'd be good to know what the pH of the water is in Salsa Creek, because the majority of our research kind of stemmed around how does the water kind of run into the soil, and that could affect it, but we didn't even know what the pH was. And also we heard a lot about pine needles dropping could affect the pH of the soil, making it more acidic. There were no pine trees that we tested. We didn't see any, so we are kind of missing that data point as well. And another thing that we wanted to maybe learn about was how is the pH of the water changed with human development, and what is the pH based off the water, like if it rains, based on the season. And overall, we kind of recognize the importance of collecting a large sample size. We had about 19 trees, but even that wasn't really enough to get super conclusive data. So if we had more options, that'd be good as well. And then also basing our conclusions off the p-value rather than the data points, because at first glance, our graphs kind of made it seem like we had conclusive evidence, but our p-values proved that completely wrong. So. Yeah, thank you. And then this is our.
All right, wonderful. Time for one question, either from the remote folks or anyone from class. Yeah, Eleanor.
Did you think that if you had done more tests in your control, you would have had like a better sense of the different soil pH levels in the trees?
Yeah, probably. We only did 2 in our control area just because of time.
Yeah.
So if we had more, then we'd probably, we'd have a larger sample size and more accurate data. So yeah.
Okay, wonderful. Thanks. Let's get our last group up. Yes.
What was the most interesting thing that you learned while doing this? Or, like, was there anything that surprised you?
I feel like honestly to me the most interesting thing was just like learning how to like test the pH of the soil, because I don't know, the process was different than I was gonna expect it to be.
Okay. Let's have the soil moisture group come up and get started. Thank you, guys.
Oh, I think it was me. Yeah. Wait, can we reload our presentation?
You need to reload it.
Yeah.
I think what they were sent, I don't know if it gets, I don't know how much of it gets.
Because I think that's the wrong version. We had some changes too that just didn't show up.
Okay. I don't know if that's possible right now. Jen, do you know if it's possible to put the most recent version, or do they? I think they're accustomed to having it. They were making some modifications towards it. Is this the one, Adele?
Yeah. Is this correct?
Great. Thanks so much. Appreciate it. All right. So it might be good. You all might need to just get going, just because we're getting close to the end of the period. Unless everyone's able to stay for the start of lunch, then I think we probably should get started. And I'll just try to hasten David in there. Why don't y'all come up to the front and just introduce yourselves?
We can just keep the last group for lunch if we need to.
Do you want your computer? Do you want your computer? Or do you want I don't. That's fine.
Okay. We did our project on soil moisture versus plant growth, and our question was, what is the effect of soil moisture on plant growth?
I'm Adele, also.
I'm Eleanor.
And I'm Devin.
Okay, so introduction part 1. So some background. What is soil moisture? So it's the total amount of water, including water vapor, in soil. And soil moisture plays an important role in agricultural monitoring, drought and flood forecasting, forest fire prediction, water supply management, and other natural resource activities. Soil moisture observations can forewarn drought or flood conditions before other standard indicators can. And then introduction part 2. So our research question was, what was the effect of soil moisture on plant growth? And then our independent variable was location. Our dependent variable was plant coverage and soil moisture or soil level. And our hypothesis was the location closer to stream will result in more soil moisture and therefore less plant coverage. And then our null hypothesis was location closer to the stream will result in less soil moisture, therefore less plant coverage.
And now for our method. So first of all, to go over how we collected evidence, we used a quadrant, which is a 0.5 meter by 0.5 meter PVC square, to mark areas to test and collect data from. And so we observed the plant growth within each quadrant and estimated the plant coverage. And we used a soil moisture tool to measure the moisture level in each square. And we repeated the process at 4 different locations, 5 feet apart. And we did this 10 feet up and down stream. And we did all of this process in 2 different locations.
So our materials, we used a tape measure, a soil moisture test tool, and a quadrant.
So to see the difference that human development has on soil moisture and plant coverage, we decided to test in 2 different areas of Diamond Park. That one is just natural, and the other one has been restored. but they were both like on the same stream and on the same side of the stream,
Right.
so that we could keep consistencies in our data.
Yep. And for our data collection, we set up tables. The left one is for the first site, the right one is for the second site, and effectively each color is a different location within the 10 feet, and in each location along the 10 feet, we measured the soil moisture at 5 feet from the stream, 10 feet from the stream, 15 feet from the stream, and 20 feet from the stream, and then added the plant coverage and moisture rating.
Okay, so site 1 again was the restored area, and our sample size was 16. So as a reminder, we used the quadrant to mark an area to test from that was 5 feet away from the stream. Then we observed the plant growth within the quadrant. Then we used the moisture tool to determine the moisture level. And then we repeated this process in 4 different locations. So 5 feet apart from, and they were 5 feet apart from each other. So 5 feet from the stream, 10 feet, 15, and 20. And then we did this process 10 feet downstream. So in site 1, we observed that the further we moved away from the stream, the more plant coverage there was and the less moisture that there was in the soil.
And then for site number 2, which was the unrestored area, pretty similar thing. So we used the quadrant to mark an area that was 5 feet away from the stream, then observed the plant growth within the quadrant. We used the soil moisture tool again and repeated the process 5 feet, 10 feet, 15 feet, 20 feet. And in site 2, we observed that the further we moved from the stream, the less moisture there was as well. So pretty similar results to the first site. And we observed more plant coverage in site number 2 and higher soil moisture levels in site number 2. So the trend was pretty similar, but the overall level within those trends was higher.
Okay, so what are the main patterns in our data? In both restored and unrestored, we saw that as we got further away from the stream, there was less moisture in the soil. And there was a significant difference in plant coverage between restored and unrestored, and it showed that areas that human development had affected had significantly less plant coverage compared to the natural areas. And 5 feet away is where we noticed that our data had the widest range between soil moisture levels consistently throughout our experimentation process, and that our p-value could not be calculated since our null hypothesis had multiple variables. So how do our results compare? Our results are both supported and rejected by both our hypothesis and null hypothesis. Our null hypothesis saying that closer to the stream had less plant coverage proved true, but it didn't stand that there was less soil moisture. And then our hypothesis, which stated that there would be more soil moisture and more plant coverage, was only true in the part that there would be more soil moisture and did not remain true about the plant coverage. And we think we saw these results because we took a large enough sample size that we were able to rule out irregularities and get a consistent ratings on each of our distances.
Okay, discussion part 2. So how did your results compare to current science and the biological context of the field site? So our results are interesting within the biological context of the field site because we took data from both restored and unrestored areas. Ultimately, we found that in the unrestored natural area, there were higher levels in the soil, whereas in the restored area, there were lower levels of moisture. How could your research affect our understanding of the ecology of the area you researched? Our research could help with our understanding of the ecology in our field site because we learned that as a result of human impact and development, there was less moisture in the soil. What could your results tell us about ecosystems like this one more broadly, especially when thinking about global environmental health? This could suggest that our climate is better left untouched and undisturbed by humans, which is an interesting approach that could possibly help improve global environmental health.
Yeah. Next, for possible future research. So to better answer our questions about soil moisture, it'd be helpful to do more research on how the soil's ability to affect water to absorb water is affected by things like light, weather, and temperature. And if there are more ways to discover an area's true plant coverage, because the way we measured it was pretty subjective. It was like us looking at what was physically there and just determining how much of the area was covered. So seeing if there's a way to do that in a more exact fashion. And learning these factors would allow us to create a set of larger and more customizable data collection sites that would take more variables into account, like I said, such as temperature, light exposure, exposure to things like rain, so that's the weather piece, and just changing the size of the collection area in order to get less biased or skewed data.
Oh, and our bibliography, we definitely had to do a lot of research on soil moisture and the effect it has on plant coverage, especially after we took our data, because we wanted to understand further why we got the results that we did.
Thank you. Any questions for our soil moisture group?
Oh, we have a question on the screen.
I think we definitely had a lot of fun. We
loved it.
Yeah,
we had some challenges due to our class schedule, so we had to kind of improvise. But I think that made our teamwork very much more collaborative.
We definitely bonded as a group.
Yes.
Great. All right. Thank you all very much. And thank you, folks, who are watching remotely. We really appreciate your participation in this new thing that we've tried this year. And thank you for your patience while we were working out any tech glitches. So, class, do y'all want to say goodbye and thank you to the audience? Okay.
Thank you.
Take care. All right. Thanks again, and have a wonderful afternoon. Thanks for watching, tuning in.